Good morning, everyone. Today we are going to discuss about Chapter 5, Molecular Basis of Inheritance, the topic genetic code. In this module, we will discuss what is the meaning of genetic code. So, the scientist George Gamow, he said that since there are 4 base sequences within the DNA or RNA, there can be a possibility of these 4 bases which can code for each amino acid. But when we go deep into the detailed study of amino acids, we have found out that there are approximately 20 amino acids are taking part in the protein synthesis in our body. And if there is 20 amino acids are there, means this 4 base sequences, their code is not enough for coding this 20 types. Of course, there are many other amino acids which do not directly taking part in protein synthesis. The main examples are citrulline, ornithine. These amino acids are not taking part in protein. But other than this, 20 amino acids are acting as the structural component of proteins. So if there is 20 amino acids are there, means 4 base, their code is not enough. Then he proposed that there can be 2 to the power of 2. That means 2 base combinations can give rise to the gene base or to the base sequences of codon. So if it is 2 to the power of 4 means the 4 bases are there. So 2 to the power of 4 means then it can be only 16. So then not that one. 4 to the power of 2. Then that cannot be enough. Then he said it can be 2 to the power of 3. 4 to the power of 3. That means there can be 4 to the power of 3 means 3 base combinations can give rise to a single, or can that triplet code can code for a single amino acid. So it is 4 to the power of n means total number of combinations which can arise from 4 bases can be, that is approximately 64. Then he concluded that since there are 20 amino acids, 64 combinations can be the possible explanations for a genetic code. So when we'll come more detail into genetic code, the definition when we say it is the sequence of triplet base sequences. Okay, it is the sequence of nucleotides in RNA or deoxyribonucleic acid or in DNA that determines the amino acid sequence of protein. So suppose there is a protein, there are say for example in hemoglobin, alpha hemoglobin, there is 146 amino acids, and in beta it is 141 amino acids. So like that amino acids, but the amino acid combination is only out of 20. 20 amino acids arranged in different pattern and they can form a single polypeptide. So here George Gamow, he said triplet base sequences can give rise to a single amino acid, or that base sequence can code for a single amino acid. So the definition of genetic code, we can say it is the sequence of nucleotide sequences within the RNA or DNA which codes for the amino acid sequence in a protein molecule. Then each triplet sequence will code for a particular amino acid. So there can be combination of 64 triplet combinations. And from this they framed the main characteristic features of genetic code. And before we move into that one, there are many other scientists also developed many techniques, and with the help of their inventions only we were able to decipher the 64 codons. So triplet base sequences in either in mRNA or DNA, we will call it as codon. So there can be 64 codons, but the meaning of codon or the each codon, each triplet base combination can give rise to which amino acid. All those things are mediated or can be brought into the light by the other scientists. Mainly the main scientist involved in the deciphering of genetic code is the first and foremost one is Hargobind Khorana. He is of Indian origin, and he invented a mechanism of synthesizing RNA molecule in defined combination of bases with the help of homopolymers as well as the copolymers of nitrogenous bases. So here he developed a technique of making or artificially synthesizing an RNA, or in vitro synthesizing, we can say. In vitro synthesizing means the making of RNA molecule outside the body, artificial. So first he used homopolymers. Say for example, U. Many uracil molecules are uracil bases linked together, and he made an RNA with U alone. And the next step he used the second type of, that is copolymers, 2 or 3 base sequences, and then he was able to create another type of bases with the copolymers, either UA or UG or UC in that 2 or 3 combinations. And he was able to make RNA strand, artificial RNA. The second scientist, he developed the mechanism of developing cell-free system of protein synthesis. That means Hargobind made an RNA, and by utilizing that RNA, Nierenberg, he developed a synthesizing machinery from an mRNA, how we can make protein outside, even that also in vitro. So as a homopolymer of U means they will make, with the help of Nierenberg's invention, they were able to make a protein strand with the U codes for phenylalanine alone in that protein. And copolymers means the combination of that 2 bases, their amino acid sequences alone will be there in the protein. Then the third scientist, he has developed an enzyme for the polymerization of these amino acids and finally converting into a protein. So that only enzyme, polynucleotide phosphorylase was the enzyme, was also helpful in polymerizing RNA with the defined sequences in template independent manner. Template independent manner means without any template. Normally, synthesis of RNA as well as the protein, they require a template, either in the form of RNA means from the DNA to RNA, and protein means from mRNA to protein. So there is a dependency of a template. So here Severo Okawo, he developed an enzyme which can mediate by itself for the synthesis of polynucleotide strand. So with these 3 scientists together, when they developed this one, they were able to find the meaning of each and every codon that out of the 64 combinations, and that they plotted in the form of a what we'll say tabular form, in a box checker box form, and that is named as genetic code. And along with this, there is one more scientist, that is Crick. Crick only has developed or he has proposed, there is in between the protein synthesis as well as the mRNA, there is a mediator, mRNA, messenger RNA, as well as the protein molecule. In between them, there is a connecting link. There is an adapter molecule. And he said there is a possibility of an adapter molecule, and he named it as transfer RNA, which helps in transferring the messages which are encoded within the mRNA into the sequences or base sequences of protein, that is called as we can call it as translation. So for the translation process, there is an existence of adapter molecule that was proposed by Crick. So all these 4 scientists, we can give the credit for deciphering the genetic code. Now we will move more detail into the characteristic features of genetic code. So here, the first and foremost characteristic feature, as we already know, Gamma already proposed, that is the codon. Codon means it's a triplet base sequence. So 3 base sequence together forms a codon, which codes for a single amino acid. So Hargovind Khorana or Gamow itself proposed that there can be possibility of 64 codons. So out of that 64 codons, 61 codons codes for amino acids. And then 3 codons which do not code for amino acids, that is called as stop codons. So 61 codes for amino acids, 61 combinations of triplets. Then second function, second feature what they said is each codon codes for only one amino acid. Each triplet will code for only one amino acid. Say for example, UUU codes for phenylalanine. And that codon is unambiguous. Unambiguous means there is no double meaning. UUU means always it is phenylalanine. GGG means always it is glycine. So in one organism UU phenylalanine, and in another organism in another it is not so. In all the organism UUU, or the meaning of UUU, always it is phenylalanine. Then the third one, what they said is they are degenerate. So what do you mean by degenerate? Some amino acids are coded by more than one codon. So typical example when we are saying glycine. Glycine can be coded by, because there are 61 codons, meaningful codons, and there is only 20 amino acids. So always there will be a possibility of repeating the same amino acids can be repeated by or can be produced by different codons. So here best example you can give is glycine. GGG, GGC, GGU, GGA, all it is coding for glycine. There are more than 4 also, 6 codons also coding for certain amino acids. So this is just for an example I said, you can take it as GGG. GGC, GGA, GGU, all it is coding for. Here there's another, it is actually GGU. GGU codes for glycine. And then exceptions, here we will say, so that term is called as degenerate. The codons are degenerate. Even one amino acid can be coded by more than one codon. But there are exceptions. Two amino acids are coded by only by single codon. So exceptions are AUG. AUG always coded by methionine. It's codes for methionine. The same way UGG tryptophan. So these two codons are non-degenerate. Sometimes they will ask examples for non-degenerate. So these are the only two examples for non-degenerate. Rest all amino acids are coded by more than one codon. The next one, the codon is read in mRNA in a continuous fashion. So from mRNA, the polarity is from 5' to 3'. So from the 5' end, the first 3 bases will be coded for the first amino acid. The same way, the second 3 will be coded for the second. There is no gap, there is no punctuation, there is no comma, etc. So it is read from 5 to 3 in a continuous manner. The first 3 for the first amino acid, next 3 for the second amino acid, like that in a continuous manner it was deciphered. Then the next point, it is the codons are code is nearly universal. Universal means it is same in all the organism, irrespective of whether it is amoeba or it is human beings. So typical example you can give, AUG always codes for amino acid in all the organisms, codes for methionine in all the organisms. The same way glycine, GGG always codes for glycine in all the organisms. UUUU, phenylalanine always in all the organisms the same. Then the next point, it is AUG has dual function. First of all, AUG is acting as start codon. So what do you mean by start codon? It gives signal for initiating the protein synthesis. At the same time, they will bring that codon codes for methionine. So always the mRNA from 5' there will be, if the protein synthesis to be taken place, means that at the 5' there will be an AUG, the start codon, which brings the methionine. Then later in the modification and or sometimes that methionine will be replaced, but always the protein synthesis starts with methionine and that is coded by AUG in mRNA in the 5' end. The same way, so hence it is called as initiator tRNA or initiator codon or start codon. And if there is a start signal, means definitely at the end of the third prime of the mRNA, there can be a stop signal. So the same way, there are 3 sets of codons which do not code for any amino acid. Instead of that, they will give signal for stopping the translation. So that is called that 3 sets are called as stop codons or nonsense codons. So these are UAA, UAG, UGA. So in an mRNA, if anywhere this 3 triplets, that is UAA, UGA, and UAG is coming, means there the translation comes to an end. That means the protein synthesis will come to an end. So all these are the main characteristic features of translation. Characteristic features of genetic code. So you can see the mind map here, the genetic code, the main characteristics. Once again, it consists of initiator or start codon, that is AUG, and the characteristic feature, the triplet codons are triplet base sequences which are present either in mRNA or in DNA, and it is linearly arranged without any punctuations or comma, etc. No gap between, and it is reading continuously. And there are 3 nonsense codons, UAA, UAG, UGA, and they are non-overlapping. So that also very important point only. They are non-overlapping. There is no overlap between. Always, every time, even if by mutation one base is deleted or inserted, they will take the first 3, then the next 3, then the next 3. There is never it will overlap. Then degenerate. Already I said that is each codon, each amino acid can be coded by more than one codon except methionine and tryptophan. Then non-ambiguous, there is no double meaning, U, always phenylalanine, G, always glycine. Then universal means in amoeba as well as in human beings, all these codons codes for the same amino acid, irrespective of the animals. Then commaless, there is no comma, no punctuation, etc., etc. Now we'll move on to 2 major terms, that is mutation. Mutation means it is the sudden change in the genetic makeup of an organism which can alter its characteristics or visible expressions. Such changes are called as mutations. So here we have to learn 2 types of mutations. The first one, frame shift mutation. So here you have to mention frame shift. What do you mean by frame shift mutation? It is the insertion or deletion of 1 or 2 bases that changes the reading frame, and their reading frame of that particular proteins in this particular translation machinery from the point of insertion or deletion is called as frame shift mutation. So it is the insertion of 1 or 2 bases from an mRNA or addition of 1 or 2 bases. So if 1 or 2 bases is added, means the entire triplet nature will change because they read in a continuous manner. So first 3, then the next 3, in that way. Into that, if 1 or 2 is changed, means the entire amino acid sequence, because the entire reading frame also will change. As a result, the amino acid sequence also will change. So that results in change in amino acid sequence, means entirely different protein we will get. So if that protein is changed, means characteristic features of that organism also will change. Such types of changes we'll call it as which one? Frame shift mutation. So here thalassemia can be given as an example for a frame shift mutation. That is chromosome thalassemia alpha and thalassemia beta we have learned. So in that chromosome 16 is alpha and 11 is beta. There mainly the change occurs due to the frame shift mutation. Then the second point, it is point mutation. We have already learned that by the example, from the example sickle cell anemia. That means point mutation means it is change in a single base pair. That leads to point mutation. Change in it. So here in mRNA, if single base is changed, the 6th position, 6th codon of beta chain, the GAG, glycine, alanine, guanine, and adenine, and then guanine again, GAG, will change into GUG. That means the middle, that codon, GAG codon, GAG codon is changed into GUG. Adenosine is substituted by uracil. in the sixth codon place, then that results in substitution of glutamic acid by valine. So only one base is changed that results in sickling of RBC within the hemoglobin. So it is a typical example for point mutation. We have already dealt with only. Then next one, it is lethal mutation. Sometimes this even sickle cell anemia, double recessive anemic conditions, that is in persons with both the traits from defective parents. One defective trait, sickled trait from father as well as sickled trait from mother, if they are getting, means surely such offsprings will die before attaining the or entering into their matured life. It is a lethal mutation. So HBA, both allelic representations are recessive, means it can end up with the death of the person. So if any mutation which can end up with death of that particular offspring or particular organism, then it is called as lethal mutation. Same way there can be silent mutation also. Silent mutation means even though there is a addition or substitution or whatever it is within the base sequence, if any changes occurs, if that change does not reflect in the visible expression of that person or in the changes that cannot lead to any change in the protein synthesis, order of amino acid sequence in the protein, then that type of mutations are called as silent mutation. So many of the mutations which are, even though there are mutation, chances are high, there will not directly reflect in the organism in the form of disorders or any other kind of complications. Such type of mutations are called as silent mutation. So one of the reason for silent mutation, it is due to the degeneracy of the codon. Here I said GGG also, GGA also, GGG, GGU also, GGC also holds for valence. So if most of the cases, the third base in the triplet, even if they wobble means even if there is any rearrangement or any change occurs in the third position of the codon, it may end up, mostly it will end up with a silent mutation. It is not going to affect the person because it can wobble by itself, and it is proposed by Crick. That hypothesis is called as wobble hypothesis. So that means the third base, even if there is any error in the codon of the mRNA, it may not end up with severe complications or any other. It will not affect the person and it will appear in the form of a silent mutation. Now we'll move on to the definition or the more characteristic features of tRNA. So it is tRNA, the existence of tRNA was proposed by the scientist, that is Crick. So Crick only said, since the mRNA is present in the cytoplasm, from the nucleus it is released into the cytoplasm and it will be bound with the ribosomes. So the codonic sequences in the ribosome and the amino acids are floating in the cytoplasm. So there is some agent which is carrying that message or which is acting as an intermediary between the mRNA and the amino acids which are floating freely in the cytoplasm. So there is one mediator or one adapter molecule which is actually correctly selecting the amino acids from the cytoplasm and keep bringing it into the ribosome as per the codonic sequences in the mRNA. So he said there is an existence of an adapter molecule, and they named it as tRNA molecule. And the tRNA molecule as such, it is developed by the scientist, Klug also. He said the Klug and tRNA molecule, adapter molecule is proposed by Crick, and scientist Klug, he proposed the structural features and all. So then they collectively they have come into the conclusion that it is tRNA is the smallest molecule. Structurally, when we are saying it is the smallest molecule, and it is soluble in nature, and it is only less in amount, concentration compared to mRNA and rRNA. Ribosomes, rRNA is acting as the major component of ribosome. So it is proportion wise, rRNA is very high, and it is acting as a structural and catalytic molecule. And the second second RNA is mRNA, messenger RNA. It is also so approximately 15% of the total RNA is coming under mRNA. And here it is only 5% of the remaining mRNA only acting as the structural component that take up for the formation of tRNA, transfer RNA. So it is also soluble, and it consists of only 10 to 15% of the total RNA production. And when we are saying that tRNA, there will be approximately 70 to 80 bases only. And since there is only 20 amino acids, the types of tRNA is also of maximum of 22 in prokaryotes and maximum of 31 in eukaryotes. And there is no tRNA for stop codons. Then the shape of the tRNA, that is two-dimensional shape, is proposed by the scientist Holly, and the three-dimensional structure is proposed by the scientist Klug. And the shape, two-dimensional shape, he proposed that Holly, it is proposed as or compared to as the cloverleaf model. And Klug, he said, in three-dimensional, it will appear like inverted L-shaped structure. Okay, inverted L-shaped structure. Then the tRNA itself is mediated by the enzyme, that is RNA polymerase 3. RNA polymerase 3 only is taking the initiative for synthesizing the tRNA from a template DNA. Then the next one, tRNA, the codon in tRNA using anticodon. So there are certain sites in tRNA. It is having an inverted L shape, and here you can observe it. So there are many special sites out there. So in this, it is an inverted L-shaped structure. So there is the beginning, it starts with 5' and it ends with the third and 3' end. And in somewhere in between the L region, that junction of these two lobes, limbs, there is a anticodon site. So what do you mean by anticodon site? Means it consists of a triplet base sequence which is complementary to the RNA. That site is, so here it is. This is the main speciality. So UCA, here you can see if it is binding the tRNA and RNA. mRNA is binding complementary. Complementary means anti-parallel in direction. So if it is mRNA is 5 to 3 means tRNA, the direction will be from 3 to 5. So in that, there is a triplet base sequence which is exactly complementary to the codon of the RNA. So if it is methionine, in the 5' end, if it is AUG, the methionine codon means the tRNA which is going to bind with mRNA in that first codon will be having AUG means UAC, anticodon will be UAC. And at the third end, it will be attached with that respective codon, that is methionine. In that way, the codon of mRNA and anticodon of mRNA will be complementary. And the codon message, the message or the meaning of that codon of mRNA will be conveyed, and it will be, as per the instruction, the tRNA will bind with a specific amino acid. So there will be a, what we'll say, there will be a meaning or the message will be conveyed properly to by the help of anticodon to the third end of this one. So if in the mRNA, if it is UUU, the codon sequences UUU means in the tRNA, the anticodon will be AAA. And at the third end, definitely the tRNA will be bringing the phenylalanine on the third this one. So in that way, there is a, what we'll say, there can be a transformation, or we can say translation of the meaning from mRNA to the codonics, to the correct type of amino acid. Then this type of amino acid, the exact amino acid only, will be brought to the mRNA. And with the help of mRNA and the ribosome and enzyme complexes, the amino acid binding as per the codonic sequences of mRNA in that will be translated in the form of protein. So here I have attached a two-dimensional structure. The same way I have attached a three-dimensional structure of tRNA. Okay, so in the two-dimensional structure, there is the 5' end. It starts with a 5-terminal methylguanosine, Mg, and there are 3 loops are there. It is actually folded in such a way in the form of a clover leaf structure with 3 unpaired loops and 4 paired limbs. And in that first limb, it is first loop, we can call it as the DHU or dihydroxyuracil type of specialized bases are highly concentrated, hence it is called as DHU loop. And that loop, with the help of that loop only, the enzyme will bind respective enzyme, which mediates a translation, that activating enzyme. Each amino acid will be bound to the tRNA with the help of a specific amino acid. So that synthetase enzyme, amino acid synthetase enzyme, will bind onto the 5' end. The same way at the second loop, which is called as anticodon loop. Anticodon loop consists of triplet base sequences which will be complementary to the base sequence codon of the mRNA. In that way, the complementary base pairing takes place between the mRNA codon and the anticodon of the tRNA. Then there can be a presence of a short arm. It is not compulsory. It can be present or it may not be present. Then there is one more unpaired loop. It is there, that is it is called as size pseudo-uridine loop, we can say pseudo-uridine loop. In that also special type of amino acid, pseudo-uridine, is more concentrated. Then it will emerge out in the form of 3' and there it will terminate with CCA terminal. So in the 3OH terminal only, the respective, based on the codonic sequence of that one, the respective amino acids will be bound. So the binding of corresponding amino acid to the tRNA, it is called as, that process is called as charging of tRNA. So when the tRNA is bound to the respective amino acid, then it is called as charged tRNA. And when this tRNA brings it to the mRNA and there the codon anticodon sequence takes place, then it will unload that amino acid to the ribosome where the protein synthesis is taking place. Then it will move away. Then again it will collect another one, and like that one by one will be loaded, and it will be assembled with help of protein synthesis machinery. So here the role of tRNA is transferring or transporting the respective amino acids as per the codonic sequences in mRNA in the site of protein synthesis. That means within the ribosomes, hence it is called as adapter molecule. So with this, we have come into a conclusion, and we will continue the modules in the next classes also, the translation and operon concept, etc. So thank you very much for your cooperation.